Hello everyone, this is Seher from EasyPeasy, and the topic that we are going to discuss today is called as oligosaccharides. If we look at the word oligosaccharides, oligo means few and saccharide means sugar. So by this way, the definition of oligosaccharide is that it may contain from 2 to 10 sugar units that are linked by a glycosidic bond. And this definition is not so strict because even if we have 25 sugar units attached with each other by a glycosidic bond, they can still come under the category of oligosaccharides. Now, if we talk about 2 sugar units that are attached with each other, then they will be called as disaccharides. We did talk about disaccharides in our previous video, so we are going to skip this part in this video. The link of the previous video on disaccharide will be provided in the description box. So by this way, according to the definition of oligosaccharide, it feels that they are quite simple. But that's not the case, because the properties of oligosaccharides depend on what type of monosaccharides are present in them. If we have an oligosaccharide in which we have repetitive units of any other monosaccharide other than fructose, then that type of oligosaccharide will be called as glycanes. But if we have a compound in which we have a repetitive unit of fructose in it, like this, so that compound will be called as fructanes. Now, why we have 2 different names of this oligosaccharides? Because they have different functional units in them. So all the other monosaccharides other than fructose have aldehyde group in them. But in the case of fructose, we have a ketone functional group there. Oligosaccharide properties also vary when we have a compound with the same repetitive unit of that monosaccharide, or we have random different other type of monosaccharides in them. The property also becomes more enhanced when we have a branch structure there and the repetitive units of the same type of monosaccharide or the random type of monosaccharides there. It starts getting more confusing when stereoisomerism is also going to affect the properties of oligosaccharide. And don't forget the alpha and beta configuration of each monosaccharide that are going to attach itself with each other to make an oligosaccharide. And guess what? All these complicated oligosaccharides come under simple oligosaccharides or free oligosaccharides. I know how you're feeling. Okay, so simple oligosaccharides are mostly present in plant cells, and they can also be called as free oligosaccharides. For example, if we talk about raffinose, raffinose is a compound in which we have 3 monosaccharides attached with each other. In this compound, we have alpha galactose, alpha glucose, and beta fructose. And 1612 is telling us that carbon number 1 of alpha galactose is going to make glycosidic bond with carbon number 6 of alpha glucose. And carbon number 1 of alpha glucose will make glycosidic bond with carbon number 2 of beta fructose there. Now, this compound have 3 monosaccharides in them, so they will be called as trisaccharides. If we talk about stachys, stachys is a compound in which we have 4 monosaccharides in them. In this compound, we have alpha galactose, and its carbon number 1 is going to attach itself with carbon number 6 of another alpha galactose, and alpha galactose carbon number 1 again going to make bond with carbon number 6 of alpha glucose here, and alpha glucose carbon number 1 is going to make bond with carbon number 2 of beta fructose. This compound have 4 monosaccharides in them, so they will come under the category of tetrasaccharide. Similarly, we have verbicose, In verbicose, we have 5 monosaccharides attached with each other, and this compound will be called as pentasaccharides. Similarly, with 6 monosaccharides, it will be called as hexasaccharide, then heptasaccharide, octasaccharide, nonasaccharide, and decasaccharides. Similarly, let's talk about the more complicated oligosaccharides, and they are called as glycoconjugates. Glycoconjugate means that oligosaccharides are not present freely in the cell, rather they are attached with another biomolecule within the cell. These type of compounds are mostly present in animal cell. So if they are going to attach themselves with proteins, they will be called as glycoproteins. If these oligosaccharides are going to attach themselves with lipids, they will be called as glycolipids. And in recent years, it was found that oligosaccharide can also attach itself with RNA, and they will be called as glycoRNA. Let's talk about each glycoconjugate separately. So the first glycoconjugate that we are going to talk about is glycoproteins. In glycoproteins, oligosaccharides covalently attach themselves with the polypeptide chain. The most common amino acids that are involved in the attachment are serine, threonine, and asparagine. The amount of saccharide that are attached to given polypeptide chain can vary from as little as few percentage to more than 60% by weight. The most common location of the attachment of glycoprotein is endoplasmic reticulum and Golgi complex. From there, they can either go into the lysosomes or go towards the cell surface. Depending on the different type of amino acid involved in the attachment of glycanes, we can differentiate them into 2 different categories. If the oligosaccharide is going to attach itself with asparagine amino acid, then they will be called as N-linked glycanes. As you can see in this molecule, the amino group present on asparagine is directly linked in the attachment of this oligosaccharide with it. And if this oligosaccharide is going to attach itself with serine or threonine, then these type of attachment is called as O-linked glycanes. As you can see that the OH of serine and threonine is directly attached with glycanes here. N-linked glycanes are usually seen in highly branched complex structures, and N-linked glycosylation usually occurs in endoplasmic reticulum. On the other hand, O-linked glycosylation usually done by Golgi complex there. These glycanes, when they are going to attach themselves with protein, they usually stabilize the structure of that protein. They can also help in the prevention of degradation of that protein by proteases. They can also control the physical maintenance of tissue structure and integrity. And as mostly they are present on the surface of the cell, they can help in cell recognition, receptor ligand binding, and cellular adhesion. The second glycoconjugate that we are going to talk about is glycolipids. By the name, we know that glycolipid will have 2 moieties in it. The one moiety will be the glycanes, and the second moiety will be the lipid. And the bond that they are going to make with each other can be glycosidic bond or non-glycosidic bond like ester and amide. Now, the glycosyl moiety can be mono, di, oligo, polysaccharide. They can be alcohol sugars, amino sugars, or sugar acids. Whereas the lipid moiety can be fatty alcohols, fatty amino alcohols, sterols, polyketides, hapenoids, and carotenoids with different substitution, chain lengths, saturation level, branch chains, and polymerization. So this is a very big topic. So we are not going to discuss this glycolipid in that detail, Rather, we are going to discuss the most common glycolipid that is present on the surface of the cell in extracellular space. Now, if you look at the lipid structure, you can see that they have polar heads there and non-polar tails there. But in the case of glycolipids, the head is replaced by the glycanes or oligosaccharide there, as you can see in this picture clearly. Let's discuss this lipid structure in more detail. So this lipid structure have fatty acid chains in the orange, and in the head region we have glycerol attached with the phosphate group, and this phosphate group is attached with the head group. If I make this compound like this, then fatty acid chains can be saturated fatty acids and unsaturated fatty acids. And they are attached with glycerol here, and this glycerol is attached with phosphate, as you can see in both pictures. Now this phosphate is attached with the head group. This head group can be phosphatidyl ethanolamine, it can be phosphatidyl serine, it can be phosphatidyl choline, or it can be phosphatidyl inositol. This compound is called as glycerophospholipid, and it makes more than 70% of lipid membrane. The rest 30% include another kind of lipid that looks like this. It has a fatty acid chain attached with another compound called as sphingosine. This sphingosine is attached with phosphate, and this phosphate have a head group there. This head group can be choline or ethanolamine. In both cases, this compound will be called as sphingomyelin. Now, the glycolipid that are present on the surface of cell membrane look similar to this sphingomyelin. What happened is there, instead of phosphate that was attached in sphingomyelin, a monosaccharide is going to come and attach itself with sphingosine here. If this monosaccharide will be glucose, then this compound will be called as glucosphingolipid. If this monosaccharide will be galactose, then this compound will be called as galactosphingolipids. This glycolipid with one monosaccharide can also come under the category of cerebrosides. If more monosaccharides are going to attach themselves with it, just like galactose with sialic acid is going to attach itself with galactosamine, and then this galactosamine attach itself with another galactose and make an oligosaccharide here, then this compound will be called as ganglioside now. Okay, now this glycolipid have various different functions. The first function is that it provides stability to the cell membrane. As it is present on the surface of the cell, so they also facilitate in cell recognition or cell-to-cell interaction. These glycolipids have been observed to play roles in regulation of cell growth through interactions with growth receptors. And glycolipids are also responsible for other activities, for example, the recognition of host cell by viruses. Okay, the next glycoconjugate that we are going to talk about is glycoRNA. It was discovered recently that oligosaccharide chain can also attach themselves with RNAs. Now, in the discovery, the RNA that was recognized to attach with glycane was yRNA. yRNA is basically a non-coding RNA, which is known to have regulatory roles inside the cell. And yRNAs are known antigens associated with autoimmune disease such as lupus. Inside the cell, this yRNA was attached with a glycane that have sialic acid in it. When these two biomolecules attach themselves with each other, they come on the surface of the cell like this and got recognized by sigillic receptor. Saccharic receptor are certain receptors on immune cells that are known to bind to sialic acid. So when this glycoRNA was recognized by saccharic receptor, as a result, it helps in the production of anti-double-strand RNA antibody. So there is a chance that glycoRNAs could help fine-tune our immune system. More research is still going on this glycoRNA. Okay, there are some dietary oligosaccharides as well, which are naturally present in our fruits and vegetables. And the majority of these oligosaccharides cannot be broken down by our digestive system. So they go into our colon, where they feed and support the beneficial bacteria that are present in our large intestine. That's why they can also be called as prebiotic fiber. Another major oligosaccharide is human milk oligosaccharide, and they are the third most abundant component of breast milk after lactose and lipids excluding water. Human milk oligosaccharides are a complex mixture of more than 200 non-digestible and non-nutritional carbohydrates, which are not present in cow milk or infant formula. And what are the benefits of these oligosaccharides are that they help in the modification of intestinal microbiota. They help in anti-adhesive effect against pathogens. They help in the modulation of intestinal epithelial cell response. And they help in the development of immune system of our babies. So hang in there, Mama. I know you are trying your best to be the best mom in the world. But if our little effort can make these little ones happy and healthy, then I think that's worth it. That's it for now. Thank you very much for watching this video. If you like it, please subscribe this channel. Thank you. Bye-bye.